Hi, welcome to Competitive Botany YouTube channel. In this video, you are going to learn important bits from microscopy. To get my next video or notifications, subscribe the channel, then tap the bell icon, watch this video until the end to get complete information regarding the topic. Please share this video to your friends, colleagues who are preparing for government jobs related to botany. Let's go to the class. Before going to the competitive bits from microscopy, I would like to give you a brief information regarding microscope. So microscope is an optical system designed for the study of objects not to be seen by the unaided eye. So it makes us see things which are very small. So it is an instrument used for clinical and laboratory studies. Okay, so this is regarding microscope. Okay, so now going to competitive bit like operational compound microscope discovered by Zacharias Janssen. Answer is Zacharias Janssen invented first operational compound microscope. So it was a compound microscope. It contains two tubes fitted one inside the other. It magnifies the object 9 times. It magnifies the object 9 times. So next, based on the source of illumination, the microscopes are classified into 3 categories. They are light microscope, electron microscope and X-ray microscope. So first is light microscope. So in light microscope, it is called a light microscope. Why? Because here the source of illumination is the light. The source of illumination is the light. It is useful to magnify the objects up to thousands or lakhs of times to the size of original objects. Okay, so before going to deal types of light microscopes, you have to know resolving power and magnification. So magnification is nothing but the microscope magnifies the object into many times. Okay, so after that you have to know resolving power. Ability of separation of closely associated objects into two separate objects is called resolving power. Resolving power of light microscope is 0.3 μm. So now let's discuss different types of light microscopes. First is simple microscope, nothing but dissecting microscope. So it is made up of a single convex lens or combination of lenses. So you can see the picture here. The simple lens is used. Okay, so next, simple lens can magnify an object only 3 times. For getting a magnification of more than 3 times, a combination of several lenses may be used. So dissecting microscope is an example of simple microscope. It is used either for dissecting the material or for less magnification, that is only up to 6 times, 12 times or rarely 20 times magnification. Okay, so it is generally used in the botany laboratories to magnify the objects. Okay, so next coming to compound microscope. Compound microscope, here it is compound microscope. You can see the picture. It has a system of 2 lenses. It is formed by the combination of 2 simple microscope. Here also the light is the illuminating source. Hence it is also called as light microscope. Okay, so next coming to the phase contrast microscope. So it is a modification of compound microscope. It is also a light microscope. It has the same resolving power as that of compound microscope. So now we are going to our topic, that is competitive bits frequently asked from phase contrast microscope is phase contrast microscope invented by the scientist Fritz Germicke. The scientist name is Fritz Germicke, who invented the phase contrast microscope. Next important bit is phase contrast microscope used to study the behavior of chromosomes in living cells. It is used to study the behavior of chromosomes. You should remember. Next, going to third question. Which microscope is useful to study the chromosomal behavior during mitosis or meiosis in living condition? Should remember the answer is phase contrast microscope. Now going to deal the fourth question. Which microscope is generally used to observe unstained living tissue? Answer is phase contrast microscope. Phase contrast microscope is used to study for the living cells. Okay, should remember it is used to observe the living cells. Okay, next used to study the behavior of chromosomes in living cells. Okay, next phase contrast microscope also used for the observation of unstained living tissue. You should remember these are the important bits frequently asked from previous exams. Okay, so next going to deal the next microscope, that is fluorescent microscope. Fluorescent microscope is an advanced type of light microscope that uses the fluorescence. It is used to study the properties of organic or inorganic substances like chlorophylls, proteins, vitamins. Okay, so next the questions that are given in previous exams from fluorescent microscope are, the first question is, fluorescence microscope is used to diagnosis of infectious diseases. You should remember for the diagnosis of infectious diseases the fluorescent microscope is used. Okay, so next question is, which microscope is useful to diagnosis of infections or diseases caused by bacteria, protozoans and viruses? The answer is fluorescence microscope. Next question, the microscope used in diagnosis for bacterial identification is? The answer is fluorescence microscope. Next question is, it is also used to study the cytoskeleton of the cell. It is also fluorescent microscope also used to study the cytoskeleton of the cells. Next question is, the fluorescence microscope is used to localize cell structures. It is used to localize cell structures. Next we are going to deal the next microscope, that is electron microscope. We are going to deal the electron microscope. Electron microscope utilizes the short wavelength of an electron beam. As the wavelength of electron is very short, the magnification is very high. So for high magnification, electron microscopes were used. Okay, so now let's deal the questions given in previous exams. Their electron microscope was invented by, answer is Knoll and Ruska. Okay, the electron microscope was invented by the scientists Knoll and Ruska. Resolving power of a human eye is 0.01 mm. Okay, so but the resolving power of electron microscope is 0.4 nanometers. Resolving power of an electron microscope is how much greater than the resolving power of light microscope? Answer is 2000 times. Okay, 2000 times greater than the resolving power of light microscope. Okay, in electron microscope, the lenses are replaced by electromagnetic condensers. The lenses are replaced by electromagnetic condensers. Let's go to the next question. The study of ultrastructure of cell and various cell organelles is facilitated by electron microscope. Ultrastructure of cell can be studied under the electron microscope. It is much useful in studying viruses, bacteria and ultrastructure of cell components. Next question. Which technological instrument have lowest and highest magnification range respectively? So answer is light microscope and electron microscope. Light microscope have the lowest magnification range. Electron microscope have the highest magnification range. Next question. Living cells cannot be observed in electron microscope because living cells, they lose water vapor and disturb the vacuum of the microscope. Hence they are cannot be observed in electron microscope. There are two types of electron microscopes, namely TEM and SEM. TEM, nothing but transmission electron microscope. SEM, nothing but scanning electron microscope. Let's see the first one, transmission electron microscope. In transmission electron microscope, electron beam is transmitted through the object, hence it is named as transmission electron microscope. Hence it is named as transmission electron microscope. Let's see the questions given in the previous exams are transmission electron microscope is used to study the ultrastructure of cellular constituents in three-dimensional views. Okay, so cellular constituents nothing but cell organelles like a chloroplast, mitochondria, Golgi complex, endoplasmic reticulum, etc. Next question is the maximum magnification that an electron microscope can give us is 2 lakh times. Okay, the next question is microscope used to study the 3D images, nothing but TEM. Transmission electron microscope can give us 3D images of cellular constituents. So next we are going to deal the next type of electron microscope, that is SEM. SEM is nothing but scanning electron microscope. In scanning electron microscope, the electron beam scans the surface of the object. It scans the surface of the object. It is actually combination of technology of electron microscope and television electronics. Okay, you can see the picture. So next, the previously asked questions from the SEM are microscope used to study the detailed account of epidermal appendages and the structures of proteins in the cells, nothing but SEM. SEM is used to study the epidermal appendages and the structure of proteins. Next question, scanning electron microscope used to study about specific proteins. It is used to study the specific proteins. SEM is invented by Manfred von Ardenne. Next question is, which microscope is useful to locate subcellular components such as specific proteins and surface view of epidermal cells along with their appendages? The answer is SEM. These are the previously asked questions from the scanning electron microscope. So next we are going to deal the last but not least, that is X-ray microscope. X-ray microscope, here it uses the X-rays for as a source of illumination. X-ray possess extremely short wavelengths and have great penetration power comparatively in thin living tissues. So hence here the illuminating source is the X-rays. Okay, so next I would like to give you a brief account on important laboratory techniques like centrifuge and electrophoresis. The first one is centrifuge. Centrifuge is an apparatus used to separate particles suspended in a solution by centrifugal force. Okay, so centrifugation is a process of separation of particles suspended in a solution by centrifugal force. You should remember. Okay, this technique is used to study breaking or lysing of cell membranes or cell walls in order to isolate cellular constituents like cell organelles and also macromolecules inside a cell. They are all separated through a process, centrifugation. Okay, so now we are going to know the important questions from centrifugation. So, which technique is useful to separate cellular components like organelles based on their molecular weight? The answer is centrifugation. The answer is centrifugation. The isolation of individual organelles from cell homogenates is achieved through differential centrifugation. Differential centrifugation. The next question is, which technique is useful to separate cell organelles from a living cell? Ultracentrifugation. The next question is, centrifugation is a process of separation of cellular components based on their molecular weight. So it's a process of separation of cellular components based on their molecular weight. So these are the previously asked important questions from the centrifuge. Next important technique is electrophoresis. Electrophoresis. Electrophoresis is a process of movement of charged particles in a fluid or gel under the influence of an electric field. Okay, under the influence of an electric field. This is regarding electrophoresis. Now we are going to deal the important questions, previously asked important questions from the electrophoresis. The first one is, electrophoresis is used to separate the charged particles. Should remember, here it is used to separate the charged particles. Like DNA, RNA, etc. Next, second question. Which technique is used to separate the macromolecules present within a cell by passing electricity? By passing electricity. Answer is electrophoresis. The third question. Electrophoresis is carried out in an inert medium. It is carried out in the inert medium. Next question is, this technique is used to separate the macromolecules present within a cell by passing electricity. By passing electricity. That's why it is called as electrophoresis. Okay. So next question is, the molecules get separated towards the electrodes of opposite charges. They get separated towards the opposite charges. The medium through which electrons enter or leave the system is called as electrode. Next question is, electrophoresis technique is carried out in an inert medium. It is carried out in an inert medium.